Scientists hope that software prediction models can help to make cassava breeding more efficient and precise so that resistant varieties can be developed more quickly. The technique is called genomic selection. In genomic selection, plant traits like yield, disease resistance, and root quality are measured and correlated with genetic information in a statistical model. When provided with a DNA sample from a young cassava plant, the software gives a prediction of how valuable the plant's genetics might be to the goals of the local breeding program.
DNA samples coming from Africa. So they send the samples in this type of tubes. So each tube contains the DNA inside. We'll send these samples to the sequencing machine. So those sequences will be generated, the sequences from each and every tube here.
We are using a tool called Genomic Selection, which basically is trying to predict the performance of the offsprings or progenies from the performance of the parents. We started this as a proof of concept to discover these tools that are needed for breeding varieties that are better than what the farmers are growing.